Hi guys, welcome to lecture 3, the third and final lecture on speciation and population genetics. So today we're going to, you can learn how we define species and how do we know when new ones are formed. And finally, why is it important to study how species formation occurs? So this illustration is of Australopithecus. It could be Lucy, Australopithecus afarensis. It could be any number of the different Australopithecines. I think there's a dozen of them. Robustus, afarensis. So there's just a whole bunch of different Australopithecines running around at the same time the Homo genus arrived. In fact, I think in Africa at one point there were 4 or 5 extant species of hominids, all more or less doing the same thing, competing for resources. So it must have been a pretty exciting time to be alive. Anyway, remember that species are groups of organisms that are so similar that they can produce viable offspring, right? And viable means that they have babies who can have babies. They're fertile. But, we can't always tell, for instance, in the case of fossil species, we can't determine whether they were able to produce viable offspring. So you could look at the bony remains or fossil remains and try to estimate species based on the differences in morphology. Morphology being the shapes of the organisms present. So how do new species form, and how do we determine that? Well, remember that mutations occur. They're the driving force for change in organisms, and so you can imagine that if you had a population that accumulated enough changes over time, they wouldn't be able to reproduce with members of their own species. And we've talked about the example of divergent evolution with our leopard populations, right? So there's actually 27 different subspecies of leopards, probably with varying abilities to reproduce with one another. All right, so there's subspecies. So that probably means they can reproduce, and maybe eventually you'd have some other new leopard type 2 that would be a species that would be unable to reproduce with any other leopards. So remember, the key is that these genetic differences accumulate over time. And of course, those genetic differences are manifested in changes in the organism's appearance and maybe their behavior as well. And one of the key concepts in speciation is this idea of reproductive isolation. So the inability to reproduce with other members of the species that you originally were from. So remember that each habitat selects on populations for different traits. And remember back from our last year, and we're going to go back and do a little ecology review next after this unit. What a population is a population is a group of organisms of the same species that are reproducing in some geographically defined area, right? So each habitat acts on different populations differently. And so some adaptations, for instance, in the jungles may not be particularly adaptive in the mountains. And when we talk about reproductive isolation and the mechanisms and how they contribute to speciation, we talk about 2 types: allopatric speciation and sympatric speciation. Now, allopatric speciation really refers to some sort of mechanism of geographic isolation. That is, you're separated in space. You can't reproduce. And over time, you accumulate changes that make it impossible for you to reproduce. Okay, so our examples are, the emergence of mountain ranges or formation of canyons, new rivers or streams. In fact, hominid evolution, early hominid or evolution in our family has to do with the formation of the Great Rift Valley in Africa. So the northern half of Africa was covered with a dense tropical forest until the Horn of Africa started to break off. It's undergone these geological changes, and it dropped down, created a mountain range, and created the big open savanna where our ancestors came from. Those primates or pro primates who were left in the jungles went on to become modern chimpanzees and gorillas, whereas we were out on the plains and seemed to be an advantage to be able to stand up in there. That's a big difference. So allopatric speciation has to do with geographical isolation. Now, sympatric isolation is a little more complicated, and so it can happen when you may not be geographically isolated, but your behaviors don't allow you to interact with one another. You occupy a different ecological niche, or your mating behaviors change, and eventually these two patterns lead to the accumulation of large differences in the DNA. And if you've got big differences in your DNA, you can't reproduce with other species like you. So again, reproductive isolation is when you can't breed with the other members of your previous species. And here's our snow leopard and the black panther. I'm going to assume they can breed because they're just different subspecies of leopards, but they're on their way to being new species. And so let's look at some examples of allopatric speciation in detail. In the Grand Canyon, we've got a couple breeds of squirrels. There's Albert squirrel and the kaibab squirrel, and they occupy the two respective rims of the Grand Canyon. The Grand Canyon has been forming, I think it's something like 30 million years, but I don't know what the detailed story of how Albert's squirrel and the kaibab squirrel have been separated. But they've been separated for at least thousands of years. And of course, they accumulate differences over time. They came from an original squirrel species, but Albert squirrel got stuck on the south rim, and the kaibab squirrel got stuck on the north rim, and you can see how they've accumulated some differences. And in fact, they are different species. They can't and don't interbreed with one another. You can see Albert squirrel's got a gray back and a white tail with a white belly, and then the kaibab squirrel has a completely different coloration, a dark belly. There you go. Allopatric speciation through a geographical barrier. Of course, another way to undergo allopatric speciation is by occupying a different ecological niche, which separates you. All right, so remember that Darwin's finches lived on these different islands. They had different environments, and they ended up driving evolution for the different species in different directions. You had finches that went on to become insectivores. You had finches that went on to become, like ground dwelling that lived off of nuts and seeds. Then you had other finches that fed on the fruits of different trees. And eventually, over time, those differences in the populations led to speciation. Now, sympatric speciation is a little more complicated. Sympatric speciation lots of times is the result of mating behavior and timing, all right? So up top, you can see this male bird showing off for the female bird in hopes that she'll like his dance there. But if she doesn't mate with him. And of course, that dance won't work with other birds of similar species, okay? So a classic example of this mating behavior and timing is modern cetaceans, whales. So there's lots of packs of orcas that are roaming the seas together, but they just don't interbreed. In fact, they eat different prey. Some eat seals, some eat squid, and They have different hunting behaviors, and they don't reproduce because they're so different. And of course, you can also have sympatric speciation occurring in the Galapagos. So there's finches there that are in the same space, but have different mating behaviors, like this bird up here. So the changes in mating behavior, or the times of the years when they become fertile, eventually leads to the accumulation of changes in the chromosomes that disallow them from having a viable zygote even, right? So the more different your DNA is from the person you're mating with, the more likely it is that the zygote forms doesn't have enough parity for those implantation and fertilization, all right? So differences are good when you're talking about breeding, but to a degree, okay? So last but not least is extinction. And why would we study extinction when we're talking about speciation? And of course, extinction and speciation go hand in hand. Remember that we've been through 7 major extinction events. We're in the middle of one right now. Right now, all the big vertebrates on the planet are becoming extinct. And the answer is what? The answer to the question why, it's because of man. But of course, in the past, you'd had events. Well, even here, we've got this or Dinotherium. I think he is. He's a rhino relative. He's part of the megafauna. Current ideas for what happened to the megafauna is that they were hunted by man, or man changed the environment. Anyway, this megafauna rhino was replaced with the modern white rhino, who's also becoming extinct. And he'll be replaced by people, by McDonald's and Popeye's chicken stands. Down at the bottom, we can see the moa, and those are Maori. Those are New Zealand hunters hunting the Maori. They hunted the giant bird into extinction. In the U.S., there was a point where I think the population of bison was down to hundreds. And that was, when the white man arrived to the West, there was herds of like 4 million buffalo running around. And they hunted them for their skins and their horns. They left them. Anyway, of course, 99% of all species have become extinct over time. So it's the nature of things to speciate and extinct, and those are two parts of the same process, right? And so the environment's constantly selecting those organisms that are fit and selecting against those that are less fit. Of course, we've got a couple more examples of human extinction process at the bottom. There's the quagga, see, sort of like a zebra horse hybrid, hunted to extinction, and then of course the Tasmanian tiger, that's down in New Zealand and Australia. I'm sorry, New Zealand, and of course they've been hunted to extinction as well. So if a species can't change quickly enough to adapt to the environment, like the introduction of a new top predator for humans, you end up going extinct. Well, that's it. Remember that microevolution is population genetics, and population genetics is really interested in studying of allele frequencies, all right? And so those phenotypic variations are those differences we see in a population. And we talked about how the normal distribution is something we saw to describe specific alleles. And then we talked about Hardy-Weinberg. Remember, this is an ideal condition. It doesn't really exist in nature, but it helps us understand how populations can change. So that's it for population genetics. I guess the next thing we'll do is have a little test review, and I'll have this vocab review for you guys done today, for you to do tomorrow. And boy, that's it. And congratulations, you've just gotten through the major portion of the content for the bio course for the year. So congratulations.